Hi, everyone.
Hello. Good evening, I guess.
Yeah, it's 9 p.m.
Hi.
How are we all? Good.
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 I
It's
thought
raining here a lot.
Pardon?
It's raining a lot, so you might hear the rain ticking on the roof later on.
That's fine. That might help, get me ready for bed. I'm surprised to see you, Wilson. I thought maybe you might have finished up before this meeting. When's your last day?
Monday.
Ah,
Next Monday.
yeah.
Yeah. So week left.
Have you got something else lined up, ready to go, or?
Not at the moment. I'm still finishing up my dissertation or thesis. It will take a bit, and I do want a little bit of a rest before I move along.
Yep.
And yeah, I mean, there's currently a lot of jobs around, medical writing, communications, and all that. It's just yeah.
Yep.
See what comes along.
Yeah.
So I heard that Hung isn't joining today.
Yeah, so we were both still at work at like 8 o'clock. But yeah, he's been doing a lot of work, doing some filming and stuff, and it's hectic. Today they started doing some filming, and then they were doing graduations at the university, so the bell was ringing in the background, so they had to change locations, and like that took so much time, and then they ran over time, and yeah, he's just like, I'm not going to make it home in time, basically. And if he stayed at uni, like he has to leave at 10 p.m. Like, yeah, so yeah, just didn't work out for him, but
yeah. That's all right.
So did the students like the presentation?
I think so. From what I've heard, sorry I couldn't be there. I was on holidays that week. Had my graduation for my PhD. But yeah, from what I've heard, they enjoyed it. We had labs on Friday last week, and I asked a few of them about that. And all the demonstrators and the other teachers who are involved in the topic said that it went really well. So thank you very much for doing it. Yeah. Did you think it went well? Did you enjoy it, or?
Yeah, it went okay, but I had a little bit delay with the train, so it was really hectic to get the computer on at the correct time. But yeah.
Yeah, I was thinking that. I'm like, oh, let's make it the easiest possible time and like push it back as far as we can. So if something happens, like we have time. But yeah, they all made that. Well, I'll give an update, but they all made their compounds last week, and we're characterizing them this week.
Okay.
And then we'll send them to you. It might not make it in time for this year, but we'll see how we go.
Does anyone know where Dimitri is to give an update on the oxemes?
Yeah, I'm not sure, and Matt's not here either, so maybe they haven't checked yet the times yet.
Oh, I was thinking maybe you could get one of your students to make the oxeme I proposed. If you have the ketone intermediate, it is just, there's a way to do it using bismuth and grinding.
Yeah, so I just have to have a student to do it. I don't actually have any of them that can work in the research labs at the moment. So because they're all just in the undergraduate labs, the work that we're currently doing.
Methyl hydroxylamine hydrochloride, isn't that toxic?
Oh, no. It's not about what they're using. They can't even go into the labs, like into that specific lab without having all of these approvals.
Oh.
Yeah.
I see that Matt joined.
Hi.
Hi.
Everyone all right?
Yep. How are you?
Yeah, pretty good. Thank you.
I mean, unless, I don't know if Dimitri's able to, I don't know what the conditions are, but Fluorochem UK, which is a supplier we've used a lot for open source antibiotics, they have a intermediate that's not that expensive that could be used to make a compound an oxime as well, that would be useful.
Hello, everyone.
Hi.
Anthony, is it the one that you proposed on GitHub recently? The one that Kimberly was supposed to make?
Yeah. Sorry.
So, yeah, I'm not sure like what's the status of that compound, like the synthesis, like in Australia. Kimberly, are you still waiting for the?
With getting a student to make it. She doesn't have anyone available. I'm gonna look into something. There's a supplier called Fluorochem, which we've used many times before, Matt, as you know.
Yeah.
So Fluorochem, we're not gonna get the exact halogen substitution pattern, but they have a pre-made ketone that's like 20 pounds, maybe.
Yeah, I actually saw it yesterday, just checked it again. So I was thinking like Kimberly, like if there are like time issues, so maybe we could make this compound like at here at UCL, because I have like the substrate, because I just made the first oxime product. And because we have like deadline in terms of the larva season, so probably
Yes.
I'm pretty sure we would be able to get it like easily. There's no problem with this.
Dimitri, if I go look through Fluorochem for those pre-made ketones, it's one step, obviously, reflux with hydroxylamine, O-methyl, you know, like how you make the...
Yeah, it's actually one of them I made Perfinox just recently, so that's not a problem at all.
It would just be me having to mine and find the ketones. So I will do that. That is on me.
Is there a particular reason why we have chosen that scaffold?
Because it was similar to the, it's similar to pyrephenox, of course, and also we've had compounds with 3 rings similar to that Hung has made, and they were okay.
Sure. Okay. It's just that
Hung,
Perfinox have 2,4-dichloro benzyl ring, and the one that you proposed has like fluoro instead of like
that's the structure that we've always been. That's the structure. This halogen pattern doesn't matter too much, I don't think. Have we seen that, Matt? Do you know?
Yeah, because we have it like in the series 1 phenarmals as well. Okay.
Yeah. Do we know if the halogen pattern matters that much?
I don't know. So wait, we're talking about the ketoxenes, right?
Well, we don't know the ketoxenes. We're talking about the triaryls, just that we base the ketoxenes off of.
I think the halogen pattern matters, but I'm not sure what we know about it. I would have to look at the composite SAR. I think it matters from memory, but I'm not sure how much we've explored it. Just before we get too involved in the conversation, just want to make sure everyone's okay with the roles here. So Hung kind of very usefully posted who's doing what at the meetings, which seems like a nice way of splitting it up. So you guys at Erasmus are hosts today, and we are minute takers. Dimitri, are you able to maintain notes of the meeting?
Yeah, I'm taking them. Yeah, all good.
And then just, you know, add those interactions. So the thing that Hung posted was really good. I have to scoot. I've been called into a meeting at 12:30, so I've got to go halfway through this. But if you're on the call, Dimitri, and can note down what people have got to do, that'd be great. So, I mean, one of the things could be to make sure that the halogen substitution pattern that's being proposed there is okay. All right. I'm going to stop talking.
Wilson, what was on the agenda for today?
Let me go to that tab. Actually, the first one is the ISNTD talk event, which Wendy, you and Matt attended, I think. Is that right?
No, we're thinking about creating it. And there was lots of enthusiasm for that across a bunch of people. There was some potential clash that we identified between this thing and something that Professor Fahal was thinking about running, but I don't think there is there's too much of a clash there. DNDi have said that they are willing to help us run this thing and arrange it. So all I've got to do now is connect ISNTD with DNDi and get them to set a date and an approximate schedule, and then we can go ahead, which would be good. So I think it would be a neat way of highlighting what we're doing. So that action is on me to connect ISNTD with DNDi.
Yeah, and I think it will be good to make it, as suggested, a broad public. And for the clinical part, I would propose Ahmed Verhaal instead of Ed Zelstra, in case Ahmed doesn't want to talk, which I think is unlikely, but then we could always get Ed to talk. But at least Professor Verhaal sees 10,000 of patients, and Ed didn't see that many, of course.
Yeah, I mean, I was hoping that we could have a focus. The ICNTD is all about kind of building communities and trying to get people involved in doing things. And so I want to try and focus the session in on what people can do if they want to get involved somehow. So try and have that as the main focus. But it would be nice to have an input from our friends at the CDC in the US. That would be good, because they don't attend these meetings, part because they're not really involved in it, but it would be nice to loop them in if we can. Seems to me. But we can work out the details later, I guess, and make sure it's not going to be a very long event. I think it's going to be a few people going to be talking about it, but just trying to make it clear to what people can do would be great.
Yeah.
Okay. So up next, should we just give a short update of what we have done on the 3 sites, Sydney, UCL, and Erasmus? Do we want to go ahead first, Wendy?
Yeah, for us, I think you can give the best update for now, but we have been working on the in vitro analysis, I guess, and MyYingi has tested the compounds against Falciparum senegalensis, the other causative agent of mycetoma. The Fennery moles seem not to be very active against this species, but the benzimidazoles, if I'm correct, they are active. So that might hint towards the next series, maybe more likely the benzimidazoles than
panic cure type drugs. Yeah. So I don't know if febendazole has had any human trials. I know it's used in dairy cows and sheep and pigs, but I don't know if there's been human trials of febendazole. It almost seems like a question for DNDi, actually.
Yeah, we could ask them. I'm not aware if it was tested in human trials, but yeah. Same for carbindazim. I don't know.
Yeah, because those are 2 agricultural chemicals that have, one of them is already used in animals, so it shouldn't be technically that hard to reposition, right, Matt?
Sorry, I was distracted by just trying to find the
So Wendy found a drug that's used in veterinary practice called Panicure or febendazole to be effective against this falsa. Is that correct? Yeah, Falciparum senegalensis, which is another mycetoma agent. So what we were wondering was, has DNDi ever looked into developing it for people?
Yeah, don't know. That would be.
That we maybe might get. Sorry, Ben can answer that one if they ever looked at it.
I'm so sorry, Wendy. Can you repeat which against which agent have you tested? Have you done the assay recently? Strain.
So Madurella mycetomatis is of course the most common causative agent, but next to Madurella mycetomatis, especially in Western Africa, Falciparum senegalensis is prevalent as well.
Yeah.
And since we
I'm just
saw
taking
when
note, that's why I asked.
So the original data and the relevant organisms are in that link I just posted in the chat window.
Thank you. So you think that so you're saying that benzimidazoles are showing like good results? Is that correct? As opposed to phenolphthaleins.
The good thing is that they were active against both, and the phenarimols were not. We tested recently the phenarimols against Falciparum senegalensis, and they were not active. The azoles were not active either, though at the concentrations test we usually test. So they do seem to have higher MICs towards CYP51 inhibitors. On the other hand, terbinafine was very active against Falciparum senegalensis, but not that active against Madurella mycetomatis. So it seems that there really are differences in susceptibility. And since currently the diagnostics is not up to date, and we need to have some selection of compound series, maybe this is a good way also to make sure that those two main causative agents are active. In Wilson's screening of the pandemic box, we also saw that when Falciparum senegalensis was active, usually the other causative agents of the Pleosporales were active as well.
And the second compound in the list is febendazole, which is a drug used in veterinary practice, and it has been around for, like, I want to say since the 50s. So this could be a possible repositioning target.
It's not sort of used for humans yet. Yeah, and although it says it has low toxicity, but it's never been sort of approved.
No,
For
which is
human,
strange. Maybe we could arrange something to just have like DNDi start working on it and be like, you know, because it would be a repositioning. It wouldn't be making a compound from scratch.
Yeah. There was a thing in Korea last year or the year before last where a doctor recommended febendazole for cancer treatment. I mean, febendazole is used for treat cancer in dogs, but I think she recommended febendazole for to treat cancer in humans, but that didn't turn out well. It was on the news. And febendazole, carbendazim, the compounds that we found were active against Madurella mycetomatis, but they're actually, I would say, benzimidazole carbamates. Those are the ones quite active against Madurella mycetomatis. The rest, the other benzimidazoles didn't.
So it needs to have like carbamate functionality. Although all of these drugs are thought to hit tubulin, as you may know.
Yeah.
So we'd have to figure out some sort of we'd have to develop it further.
Yeah.
Febendazole might not be the.
So we have the data of the few benzimidazole carbamates that was active against Madurella in the data is all on GitHub.
One of them is actually used in people. Mebendazole is. Yeah.
Yeah, that one was not active, if I recall, Wilson.
No, I don't think that's a carbamate.
No, it doesn't have carbamate, but it's listed on there. Hang on.
Yeah.
Oh no, it's listed on the top, but it's the least potent. Okay. Sorry, I lost track.
Just trying to find out what the structure at 387 is, the other one. I probably should know that. The two that were significantly active.
I think it was, I think one of them was a methyl analog of carbendazim. It was that MMV compound, one of these MMV compounds I remember looking up, and it was actually a methyl carbendazim where there was a methyl ring attached to something to attach the benzimidazole core itself. I had it. You had some stuff listed down, a couple agrochemicals. MMV 677 was a TB compound, and it's a benzimidazole. It's a methylcarbendazim derivative. So these are all derivatives of carbendazim. So carbendazim is basically the lead.
Okay, so fenbendazole, not flubendazole, right?
Yep.
But
yeah,
Our lead right now for the benzimidazoles is carbendazim, if I'm not mistaken.
For bendazole, carbendazim, and the MMV compound that I just put on the chat, 1782387.
Yeah, I've got the structure of that.
If you could push that, that would be good. But
So it's these two.
yeah, it is carbendazim. That
These are the two
MMV,
that are active.
that is carbendazole. Was it?
That's
I
the
guess-
MMV compound.
Yeah, that's carbendazim with a methyl group. That is.
I guess the question is whether or not this motif here is required. So in other words, are there any inactives with this in them? It would be useful to know if this methyl carbamate is, well, this basically what's in the box there, whether that is necessary or sufficient. That would be useful to know. And the only reason I'm asking is because the other structures here that are shown on on the list here in series 5, I'm just not sure if this is all the structures, then we already know this, I suppose. But I guess DND might be able to advise if there are any other compounds that they have with that motif in them, I suppose.
Yep, they can. Yeah.
So a little bit of analysis of these data, because you've got things there that are. So albendazole has the same motif, but is I'm not sure which is albendazole.
Yeah, these were the ones I found on this paper of the pathogen box data. So because I'm not a chemist, I cannot make those structures from smileys because
Yeah.
I simply don't know how to.
Sure.
That's why I copy paste all the figures they gave in that paper underneath here.
Right.
Albendazole and thiabendazole wasn't tested. It was not in any of the boxes.
Okay, it would be useful if we could assemble a diagram that collates the table with this chemical structure and combines that with the activity.
Also potentially useful to try albendazole because that has a very long history of eumycetoma.
Is this also only the pandemic box? Are there any of these similar compounds or from the same family in any of the other boxes we've tested that we can see if they were active as well and maybe pull some more information?
So
yeah, Wendy, you can go ahead, please.
yeah, so Kiran Deep of MMV gave us a list of the compounds from the other boxes which we tested. And Wilson is currently making that into a table or figure.
Okay. Albendazole. I don't know if you can get it from the hospital you're working at, maybe. If they compound it, albendazole isn't usually that hard to get.
So I think digesting the data here will be useful. So what's active and what isn't would be useful, and then thinking about whether or not we have any compounds available through DNDi or someone else that has the motif in it. I reckon those are the actions there.
Unless Wendy could request a sample of albendazole from DNDi.
Yeah, that's what I mean.
Yeah.
I have attached a screenshot of the table that I've made with all the benzimidazoles in all 3 boxes, pathogen status and pandemic box there in the chat. And then the next one is just some other information. For the pathogen and pandemic boxes, we are able to get the structures from DNDi and they're readily available. But for the status boxes, DNDi is unwilling to give us because I think
MMV.
it's classified. Sorry, MMV. Yeah.
Classified.
Or they're not allowed to just give it just for the status box.
Really?
Yeah.
Do
Strange.
you have the smileys or something for them then?
I don't know.
We can definitely make, like, I can easily copy the smileys into ChemDrill to get the structures.
No, in the status box, the smileys are not given because otherwise it would be not that difficult, I guess, to these were compounds still in development or halted. Therefore, this box was not given to everybody, but we were given a copy, and we were lucky with that compound you're working on, so the aminothiazole, because they were able to disclose the information behind that compound. But for some of the compounds in the box, they are not able to disclose all the information yet. So it really depends on which compounds you want information for.
Yeah. So I've asked for all of the structures from the stasis box for the benzimidazoles. MMV said we can only get the 5 or 6 of the most active ones.
I get, at least we can get that.
Interesting.
Interesting.
Okay. I mean, I guess just the correlation between the structure and the activity would help us to ruminate on what's needed.
Yeah, or also maybe getting albendazole and screening it if Wendy is able to, and Wilson are able to.
Yeah, I can look into albendazole to see if either our own pharmacy has it or if it's not too expensive to buy from Sigma.
But I guess just to follow up the basic point here, though, Wendy, is the fact that they're active, some have some activity against senegalensis means that you are more interested in those molecules generally.
Yeah, because what I'm afraid of course, we already spent a lot of time on the phenerymols, but if in the end we end up with the perfect phenerymol and it is not active against the other causative agents, then it will not move further down the line anyways. So maybe it's good to focus on those which are at least active against a causative agent, which is also very prevalent, but then maybe in Western Africa. Although Madurella mycetomatis is still the very most important causative agent, and it's still very important to focus on that. But just as a precaution, let's include Falciparum senegalensis screening as well, just to make sure that the compounds we identify are active against that one as well.
Do you have other fungal isolates, other species like white
yes,
grains?
Yes, we have.
Because we could screen pheneromols against that, could we? Pick the most active pheneromol?
Yes, for now we screened against Falciparum senegalensis because what we found out, well, you can roughly divide the causative agents into 2 groups. One are the Sordariales, where Madurella mycetomatis belongs to. The others are the Pleosporales, where the majority of the other causative agents belongs to. So that's a little bit comparing human to fish. And what we saw was that Falciparum senegalensis behaves roughly the same as the other causative agents belonging to the Pleosporales. So that's why we selected that one, because it was the most common of that group. We have multiple isolates of that one, but we do have isolates of the other ones as well. Metacopsis romeri also belongs to this group, but in general, it's very resistant to any antifungal agent. So far in the screening Wilson did, I think we found 4 or 5 compounds with activity against that one. But because it's so very rare, maybe that should not necessarily be a downside if a compound doesn't inhibit that one. Because in literature, I think only 5 or 6 patients were identified with this fungus.
Yeah, it reminds me of the other one too, Boydii as well, very rare.
Scedosporium.
Yeah, that one is more common, but it's more common in more temperate regions. So it's causing white grains. So even if your diagnostic performance is not very well, you can at least discriminate between white and black in terms of the grain. So then you could already have some identification or discrimination if a drug might work, yes or no.
Do you think it's worth trying the pheneromols against Boydii?
We could try, but I don't have, because I have many black grain causative agents, I don't have Boydii myself, but I can ask the Fungal Biodiversity Center if I could get an isolate from them. But then I'm not 100% sure if it was from a mycetoma patient, but at least they have Scedosporium boydii.
Yep.
Once you've collated all of the structures or even just that table, I'm happy to have a look at that, and I can maybe try and look at some of the structure activity and work out what sections we think are important and what aren't.
Sure. Yeah, that's good. I think that's it for us from the Erasmus. Anything from Dimitri, Matt, or Kimberly?
Yeah, I can share a little bit from the series 2 work. So we've been working with our first year undergrads. They finished the synthesis last week and we're doing characterization this week. I'll share my screen so you can see the compounds that they have made.
Dimitri, I posted the ketone intermediate to GitHub.
So basically we have the hit compound at the top, and then as part of the training, the demonstrators have also done this synthesis. So we got them to make their own compounds as well. So we've moved the nitrogen around the ring and removed the nitrogen. And then the students have worked on these other 10 compounds. So we'd already done the nitrogen at the 2 and the 3 position. So we've moved it to the 4. And then we've done substitution at the 2, 3, and 4 positions with chloro, fluoro, and the methoxy. We think they've made all of them just based on what we've looked at so far, but we haven't done complete characterization for them. So we'll
awesome.
so we'll have all those samples hopefully characterized on Friday, and then we can ship all of them off for testing as well. So
I guess this is pretty rapid, like click type chemistry.
It's just substitution reactions between an alpha bromoketone and the thiourea.
You get a nice solids.
Yes, and like most of them don't even need to be purified. They're very good.
And the colors you posted on that Twitter
Yeah,
photo,
they're
are they
not-
real?
They are real, but they're not the compounds. Basically,
Are they underneath?
Yeah, they filter it out, and the color goes into the filtrate, and their compounds are just like normal, what you would expect, yellows and
We
white.
should try and characterize some of those colors. We could make a new set of dyes.
Well, we threw all of that out, but yeah,
Yes,
we should have.
indeed.
It was still cool though, because that was all one group as well. So for some reason that group just had all the colors and the other groups didn't, and there was almost like a whole rainbow. So
That's really good. Do you know how many of these compounds are new? According to sci-fi and that kind of thing.
yeah, so some of them are, particularly because they have the 4-methyl group on the red side of the ring there. A bunch of them have sort of been made with the 2-pyrrole group, or just without the nitrogen at all. But with that methyl group, a bunch of them are novel, but some of them also aren't. So
I have a very silly proposition, but have we attempted to methylate the hit compound of that nitrogen? The piece in red?
The
you want
NH.
the actual nitrogen,
Yeah, the
the
allopathic,
amine, or the one in the ring? Sorry,
the amine functionality.
Yeah, so we
That-
haven't. So basically, these are the first ones we've done, and we've looked at making changes to the green structure, and then we're going to look at making changes to the red one, and then we're
Yeah.
going to look at the 5 positions. So in the green, in the aminothiazole ring, substituting that, so you have 4 and 5 substitution. There's a lot of hydrolysis of this compound in the liver, and to avoid that, you can substitute at both those positions. It helps with that. And then we can also look at what linker
I was
we
just
have
saying,
in there.
shouldn't it just be methyl iodine and a base to N-methylate that hit?
Yes.
So maybe.
Yeah. The other thing is though that like these are first year students, so that's something that we would have to do ourselves. They can't do that, and it has to fit within their lab program, which is over. So that's for the another someone else to have a go at.
Yeah, it's true. Alkyl great.
That's fantastic.
When we received
Sorry,
the-
sorry, I have to duck out. I'll follow up with anything on the GitHub. If there's actions I need to do, just please put them there. But that's really great, Kimberly.
All right, see you, Matt.
So Kimberly, we will screen them against both species when they arrive.
Cool, that sounds good.
Do you have any plans, like when you would like to send them?
Yeah, so if we get all the characterization done on Friday, like I just want to make sure that they actually are what they are. And we're going to do HPLC as well. So I want to check the purity because some of them we have just got the solid and we haven't done a column. So we might have to do a column if the purity is less than 95% or something. So I just want to double check those things. And then if that's all good to go, like I'll ship them straight away.
Are you generally purifying compounds by HPLC?
No. We're just doing that as a characterization so the students get to experience that. Okay. But then because they're doing that, I can look at the purity of it and I can determine whether or not I need to do a column or not.
So they basically synthesize the thing, then they do the normal purification, like as usual.
They precipitate it out of solution.
Also, it's like a pure solid usually.
Yeah,
Oh.
yep. And most of them look pretty good from the NMR, but when we look at the mass spec and the HPLC, then I'll know more about the purity. But they look good, so.
Just last question. So coming back to the that oxime that Anthony proposed. So are we clear? So are we okay with UCL making that one? Because
yep, go for that because I don't have any of that ketone left because Hung and I have both been using that for other things. So I have to carry through more material, which is why I haven't done the synthesis yet.
Okay.
Like
I also, yeah, that ketone would be the best one, just using that substitution, that fluorochloro or fluorobromo substitution pattern. That should be from a Grignard, then an oxidation, then making the oxy.
Yep. Yeah, that's what we do.
All right.
I think I can proceed like with the UCL update now, if you're done.
Yep.
Yeah?
Yes,
Yeah.
thank you, Dimitri. Go ahead.
That would be fantastic if you make that compound. I feel very honored.
Okay, so a quick update like on the general chemistry. So first of all, one of the recent compounds that I've made, well, not actually made, but like deprotect the DM23/6 phenomenal that we have already tested. So now we have a deprotected version as well. Is a TFA salt. So that's like the latest addition to our library. So it would be interesting to see like how it performs, because just a quick reminder, that one, with the protected group was potent like at 100 micromolar, but then it lost its potency like at the 25 concentration, 25 micromolar. So we still need to just make some conclusions about that compound, like on what sort of results it will generate like in this altered version. With regards to oximes, so basically I made the ketone intermediate, and then I used bismuth oxide to make the final oxime compound. And probably I'll be using the same procedure with the fluorochem substrate as well. So it was okay, but the synthesis is not great, to be honest. I don't know, like I get lots of, so for the ketone, I get lots of benzoate unreacted, so the yields aren't great. The second step works perfectly well, but there were like some purification issues, so I had to just put the semi-pure mixture in the final reaction to get the product. But it converts perfectly well. It's just like I didn't know exactly how much. Well, now I do. I didn't know exactly initially how much I was reacting. So the yield is like not great as well.
So I posted various different things. There's 2 commercially available ketones, and then there's the one that I originally proposed that you would have to make from scratch.
Yeah. So that's with regards to oximes. Coming back to phenomenos as well. So we were thinking about like what, based on the results that we got from the screening campaign, like from the last screening campaign. So DM7.1 was is now the second most active compound, like in vivo, from the entire library. So I thought like maybe we can elaborate on this a bit more. So with what we would like to propose is like one of the new analogs probably would be the pyrrolidine as well. I also thought maybe to elaborate on DM7.1 and propose like this substrate as well. Though Matt was like a bit skeptical about this because he said like the nitrile apparently is too far away. But again, like we don't know the exact mechanism of action, so maybe this should still go. Though I don't know the exact log D value because we need to ask Ben to generate this with the start drop software as well. But we would definitely do the pyrrolidine as the latest edition as well. So we did like a bunch of stuff. So we did like heterocycles, we did fused drinks as well. I don't know. Also, there's one compound from the mega threat that probably I would quickly make as well. So it's that one. Probably with a Buckwell heart recombination or something like that. Yes. So, and basically in terms of like the what we have like in total, like for the upcoming screening campaign. So those compounds like highlighted in green are ready. DM27 and 28 has been synthesized before, but it hasn't been tested in the previous screening campaign. I'm not entirely sure whether it's in Rotterdam or it's still in the lab. I will double check that. But those are ready. So by the end of by the 1st of July, we'll have like some extended like library to test in addition. And we will make some additional decisions on what to make next with the oximes as well. With that final reaction. Because we aren't quite convinced with the synthetic route. So maybe we have to redesign it again. But the final step works perfectly well, so I'm pretty sure we'll get like enough.
Yeah, because there are some commercially available 3-pyridyl ketones, as I've posted.
The problem here was that we didn't. I couldn't find like this specific ketone, like commercially available, so I had to make it myself.
Yeah.
So I did like a bunch of stuff to do the modification, but it didn't help. So I don't know. Maybe we should redesign it again. But we'll see. But the point is, like, I got the final compound, so that's the most important thing.
Well, Fluorochem stocks a compound with that methyl removed, so it's just the 2 rings, and then it's 4 chloro on the ring. So good order, try that. And I would save you. It wouldn't be the exact compound I proposed to Kim, but it would be close enough to it that I think it would be fine just to order that from Fluorochem and do the final step with the bismuth.
Yeah, no problem, because we have like lots of the substrate, so it wouldn't be a problem.
Yeah, so it's like 30 pounds, but it has a lead time of 2 weeks, it says.
It actually comes much quicker, like in the UK.
So
it wouldn't be a problem.
It depends on. I guess we'd be able to get it done. Yeah, get it done before larvae season.
But again, like Wendy mentioned before, that probably phenomals like at this stage are not the best option because they're not active against multiple strains. But still, we would like to maybe slightly elaborate like on this active that we had like from the UCL screen, from the UCL library, sorry.
Yeah, we said that this year we would round up the phenol, so anything which is still offered this year, we will, of course, screen.
Yeah.
So hopefully that, and then maybe we can get a sub-series, I don't know, oximes.
Yeah,
It's
so
not
we'll
much.
have like in this screening, in the upcoming screening campaign, we'll have at least like 2 oximes, but we will have a look at the Epichem library again to see maybe we can make something more, just to see like the active motives and so on. Kimberly, any thoughts about the carbonitrile? What do you think? Just wanted to ask your opinion on this.
Yeah, I think that this will be interesting anyway, because I think we've looked at like with a phenyl ring and the nitrile on it, so seeing whether that aromaticity is important or not will be interesting.
The only problem we have currently is I don't know the log D, because Ben uses start drop, and we agreed that we would use the same software, because different softwares like generate like there's a deviation like in the results. So just like for consistency reasons, we need to. I'll probably ask him.
Yeah. I think it's okay though to get like an estimate to see whether it sort of fits in with the trends that we're seeing and whether it's a compound we want to make or not, and then like getting the exact one. I think it's okay for now, and then you can, yeah, like if you decide you're going to make it, then you can ask him. Because we can't ask him every time we come up with one structure. Like, but yeah, I think it's worth it.
Yeah. Because
the oxenes as well. So that intermediate that you need for the one that has been proposed, that it won't take you long to make it either. I'll share my ELN pages with you, but the Grignard's like 1 hour, and I do a tempo oxidation, which takes like half an hour. So it shouldn't take long to make that. Pardon?
For which one are you talking about? The
for the one that Anthony has suggested.
Oh, okay.
That I was going to make.
The original compound before I started.
Yeah. 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 So if the lead time is really actually 2 weeks, you could make it in a day.
No, it's not. That was actually not even the right one. That was a different compound.
Oh, okay. Okay. Yeah.
But I think it would be the best to just start with the original one if it's that easy.
Yeah, it's quite quick.
So you could just, I guess, write up a synth root for him, you know.
I can just share my ELN pages on GitHub.
Yep. So probably that's it from us. Yeah.
Dimitri, I mean, on the agenda, it says, have you looked into the duplicate compounds with different?
Yes, I did.
Okay.
There was actually only 1 or 2 compounds that like Hank mentioned about like previously before the meeting, but we resolved like straight away because I added like where it was racemic mixture, I added like the comments.
Okay, thanks.
Brutanox, by the way, is going to be racemic as well.
Yeah.
Mixture.
Will you be sending some compounds to us, Dimitri? And do you know when?
Yeah, that's exactly like actually that was the main point. So we planned the whole work based on that deadline. So I've been keeping like that 1st of July before the 1st of July deadline in my head. So yeah, so that's why like I took like for instance like 1 compound like from the mega thread like from the master list, and we proposed like 2 new ones. So definitely want to make those at least.
Yeah, got it.
So currently what I've been doing like in the last week is just like restocking on the aryl halide and doing the piperazine scaffold as well, because I don't have that one. But then it should be absolutely a quick process. So I'm pretty certain like that we'll get those compounds in time. Okay. And the green ones are ready. Even the first oxime is ready. Just finalizing the characterization.
Yeah. Okay.
So they can go like.
Yeah. I guess it would be make the second oxime, then ship all the lot.
By the way, any comments, like any preferences, would you like us to send them like even earlier than initially planned?
Yes, the earlier the better. So I mean, just so that
Shall we
if
split it like in two parts then?
yeah, great, because it's better to do that rather than we do a big batch in one go. That
Is there a preference
minimizes.
when you would like to get like the first batch?
Sorry,
Is there a preference like when you would like to get the first batch?
if you have it ready, you can send it to us whenever it's
Mhm.
ready. Yeah, just so that we don't test a big batch at once in the end. This minimizes errors on our side.
Okay, okay, we'll consider this. Then I'll just wrap like the characterization up and
Yeah, and if there are good hits, then we can screen because the larvae season already started. So if there are good hits, we can proceed those hits already earlier to the larvae season. Yeah, that makes life easier. Because now, last year we had a very warm season, so a very nice long larvae season. But currently we still have cold temperatures, so it's probably a short larvae season this year. So the more we can do early on, the better.
okay, that's good to know because we see actually we were planning like by the end of June, but probably we can send those, at least the ones that we got. It's not a problem. Yeah. We'll just double check that they're stable. We'll do the NMRs quickly just to see that everything's okay, and then we'll send them.
Sure, thanks, Dimitri.
Yeah.
I think, I mean, do you have any updates, more updates on your side, or is that it?
That's it. I think that's it.
Okay. Do we have anything that we would like to, other topics or subjects that we would like to touch upon for the next meeting, for example? Because I don't think we have much tasks for the next meeting.
I've just noticed the final one on the agenda is like Caitlin to look into the SAR stuff. I'll get in contact with her and see how she's going with all of that as well. Because we were chatting with her in the beginning, and yeah, I'll see how that's coming along.
Yeah.
Okay.
Great. Anything else? We have 10 more minutes left. If you have anything, you can questions, whatever. Just if
I asked this before, but I'm so sorry for asking this again. Is there a reason why DM25 didn't proceed to in vivo? Because it had like those like relatively good values. So for example, like minimum inhibitory concentration was the same as like with DM7, which is like the second most potent compound. I was just wondering, like, was there a reason?
The only reason that DM25 didn't go through into the in vivo stage was because we had quite a large batch of faint animals from UN home to test, and a lot of them had an MIC50 value of below 8. So it was just a cutoff value that we have to put. And if we included 8 MICs of 8, compounds with MICs of 8 in there, then I would have too much to do in the larvae season last year. It was that.
So the cutoff point was MIC for MIC50 was
yes.
8.
Yes.
How then like DM7 went through to the next stage?
So DM7 was also, we have tested DM7 twice, yeah, because it was also P4C003, and that had an MIC of less than 8. So we thought, oh, okay, they are the same. We'll just use your DM7/1 name for that. If you think that we should test DM25/1 in our Vivo, then we can do that. I mean, you said based on the structures, right? Because they're quite similar. It's quite similar to 7 and 10, which had a okay activity.
No, it's just like it gave like good results both like at both concentrations in vitro. Maybe it should have.
Yeah. So
because like if you look at this graph, like all 3 molecules that are involved, like 7, 10, they were like the most potent actually, and even like 10/2 had like the lowest, like a lower minimum inhibitor concentration.
Okay.
Yeah.
Would you like
Yeah.
us to test it in vivo? Sorry, really.
Yeah, that's because here you only display the 125. And if you look at your table, that's quite consistent where the MIC 50 would be. But of course, it's not only this compounds we screen, it's the whole picture of all compounds. And therefore, only those with an MIC 50 of below 8 were chosen. If we are going to screen this one, then I think because it needs to be logical anyway, it needs to stay logical, then we need to see how many other compounds have an MIC 50 of 8, because then we would need to screen all of them, because somewhere we need to set a cutoff value.
See.
So besides DM25/1, there's only one other compound that has an MIC of 8, and that's Hong's compound 28-1F2. I don't know the MyOS number, but that's HPD 28-1F2. So
yeah,
2.
so we could consider that, but maybe it's best if you could send us if you still have a powder form of this one, so that we can redissolve it fresh, because it has been in the -80.
DM25, you mean?
Yes.
Yeah.
Because there's also like one of the non-tested analogs. So we elaborate like on this one actually by extending like the ring, the hydrocarbon chain slightly. But this has
Okay.
not been tested yet, so it's like interesting to see like what sort of results you will come up with, and maybe then we can decide like whether it's worth. Okay, we will have a look at that as well.
Yeah, and if you want us to test 25 again, please send us a new batch, because
28
we
is
have
the
found
5, but it's slightly extended, so it's like very
Yeah.
similar. We just want to see like what happens.
okay. That's fine with us. Just that if you want us to retest again 25, to send us a new one, because we are not sure if our if the one that we currently have will work again, because we have troubles
Okay.
with stability after a while.
Yeah, probably that's it then.
Anything else? Any more questions, inquiries?
No, I think it's good.
Okay.
Spot on.
So
So
then,
yeah,
so next time, again this time, that's correct, isn't it?
yeah.
Yes.
So
Okay.
no more 10 p.m. Meetings anymore.
No, it'll be this time for the foreseeable future until daylight savings or ruins us again.
Wilson, will you be attending the next meeting, or that's your last one? Because you mentioned something.
This is my most likely my last one. Yeah. Well, I mean, I don't know still for the paper, but it will be most likely my last one. So I would like to take this opportunity to thank you all for the past year of all the fun meetings and the exciting work we're doing on MycetOS. Thank you very much. And yeah, I hope to hear from you guys again soon regarding the paper or anything else. Yeah. Thanks.
Thank you for all your input.
Thank you.
It's been great.
Thanks for the work. It was nice working with you.
Oh, thanks, guys. Okay, I think it's it for the meeting today. Have
Everybody.
a good night, Kimberly, and for the rest of us, have a good afternoon. Bye.
Okay, bye.
Bye. See you next month. Bye.